there is a dynamic interaction between harmful algal blooms, cholera outbreak caused by VIBRIO cholera, and climate change. How? So increase of temperature increases the internal phosphorus loading from sedimentation and rock erosion in the water bodies, plus external phosphorus loading that comes from agricultural industry such as the use of fertilizer. So this phosphorus feeds the blooming of herbs, which algae now becomes a vector for VIBRIO cholera to populate. So we propose the idea to control this phosphorus concentration in water bodies through activated charcoal in adsorption mechanism, but this activated charcoal is made from coconut shell. So this study is going to focus on the effect of phosphorus removal in the constant water temperature on VIBRIO cholera population, and to investigate the efficiency of activated charcoal made from coconut shell to remove the phosphorus. So the presence of VIBRIO cholera in the sampling water that is going to be taken around UNIMAS will be quantified before and after the experiment. So we hope for less VIBRIO cholera after adsorption, and coconut shell activated charcoal is a practical phosphorus mechanism in aquatic system. So some might wonder why we choose coconut instead of other plant or waste. So it is simply because Malaysia is very rich with coconut, and coconut is easily and readily available almost everywhere. So who knows that this can boost the coconut industry to produce more activated charcoal from coconut shell.
So hello, good afternoon. So today I will be presenting the introduction part of this proposal. So this introduction part also includes objective and problem statements. Okay, what is VIBRIO cholerae? VIBRIO cholerae is an immoderately motile and a gram-negative bacterium. They can be found in aquatic environments such as freshwater lakes, rivers, coastal areas, and contaminated water, where these species experience fluctuation of temperature throughout the year. They can survive in both salt water and fresh water. So also, VIBRIO cholerae can cause a disease such as diarrhea in human body, which is very dangerous. So what is the correlation between VIBRIO cholerae and harmful algal blooms? Since this is our main focus on our proposal. So they both grow exponentially together depending on the environmental factors such as the level of salt, which is phosphorus, and the temperature. Next is a previous studies has been found by a group of scientists in Bangladesh in 1990. They found that VIBRIO cholerae lives in the mucilaginous sheath of cyanobacteria. And lastly, both of these organisms are favorable in aquatic environment, such as salt water and fresh water, and they reproduce well if given enough essential needs. So moving on to the next part of this presentation, which is the problem statements. So there are basically three problem statements we stated in this proposal. First is the effect of salt removal, which is the phosphorus, in the constant water temperature on VIBRIO cholerae population. Next, our concern is whether activated charcoal that is derived from the coconut shell is effective enough to remove the phosphorus in the component of water. And the last problem statement is as fluctuation of temperature occurs throughout the year, together with the supplementation of phosphorus to the aquatic environment, it is obviously will directly affect the VIBRIO cholerae and harmful algal blooms together. So our concern is whether it will increase the risk of association between VIBRIO cholerae and harmful algal bloom, as both are mutually associated with each other. Okay, next is our objectives. Basically, we have two main objectives for this proposal. So first is we would like to study the effects of temperature increment and low phosphorus concentration on the VIBRIO cholerae population. And the last objective is we would like to investigate how effective is it to use activated carbon that is derived from coconut shell to remove phosphorus in water bodies.
Okay, so for the methodology, the location of the research will be conducted in UNIMAS, specifically the living dorm of students in 7 different colleges. We will collect 40 water samples, where 5 water samples from each collection point. And collection of water sample is to be done as quickly to minimize the variation. After collection, the sample bottle is immediately sealed and transported in a cool box to the lab, and 10 gram per liter of alkaline peptone water is added as an enrichment medium alongside 20 gram per liter of sodium chloride, and then incubated at 25 degrees Celsius for 24 hours. And then, after incubation, to identify the presence of Vibrio cholera, we will use Analytical Profile Index 20E test strip by culturing the isolates in a nutrient agar. And inoculation of the test strip in 0.85% of sodium chloride of saline water, and then incubated at 37 degrees Celsius for 18 to 24 hours. And then further distinguish using species-specific OMPW-based PCR method by using the phenylchloroform DNA extraction method. And next, to test the effect of low phosphorus concentration of Vibrio cholera, we will use activated carbon from coconut shell waste. First, the coconut shell will be carbonized at high temperature of 1100 degrees Celsius in steam for over 12 hours, followed by cooling for 12 hours. The initial phosphate concentration and final concentration is calculated by using Beer's law to calculate concentration, where A is absorbance equals to molar extinction coefficient multiplied by concentration and path of length of 1 cm. The absorption of phosphate by activated carbon is aided with lanthanum oxide that acts as an absorbent. The pH of the water is kept constant at range of 5.5 and 7.2. Then, to test the temperature will be measured at 30, 40 and 50 degrees Celsius by heating a water bath at the...
Assalamualaikum warahmatullahi wabarakatuh. And my name is Muhammad HAYDAL bin Muhammad SHUKERI, and my matric number is 64941. My part, I will explain about expected outcome in our proposal. This study explains the temperature and the low concentration of phosphorus do affect the Vibrio cholera grow. Vibrio cholera show that there are modifications in the transcriptome as it a sign of the change in the temperature during cold temperature. Moreover, low concentration of phosphorus affect the growth of Vibrio cholera and algae, as a phosphorus is their limiting nutrient to survive. Regarding phosphorus, this study also is conducted to investigate the efficiency of activated carbon derived from the coconut shell to absorb phosphorus in the water body. This will help to reduce the constant supply of phosphorus to the water, which is the source for Vibrio cholera and algae to survive. This idea of using coconut shell-based activated carbon, which is CNS, indeed useful to absorption efficiency is a great due to various functional groups present in it. This study provides a better opportunity to learn about environment factors such as the temperature and the concentration of phosphorus do affect the Vibrio cholera and algae on their survivability. Also, this study will help to generate income to the country and offer the better insight into the Vibrio cholera mechanism. That's all for me. Thank you.